Role of magnetic field and Brassinosteroids in mitigating salinity stress in tomato, Lycopersicon Esculentum L. Abstract. This study was carried out to assess and evaluate the treatment of tomato seeds, var. Lycopersicon Esculentum L, with magnetic field, MF, and exogenous foliar application of 24-epibrasinolide, EBR, hormone on amelioration and alleviation of the harsh impact of salt stress on tomato growth, yield and biochemical parameters. A factorial experiment was designed with 3 replications. The experiment was carried out in pots inside the greenhouse of the Faculty of Biological Science in King Abdulaziz University, in Jeddah. Plants were watered with saline water with 50-150-250 mm, NaCl concentration. The seeds were pretreated with static magnetic field, SMF, with strengths of 50-100-150 mt, for 1h, period of time. The EBR hormone was applied as foliar spray on the leaf surfaces of tomato plants at 3 levels, 0.5-1 to 3 m. The foliar application of EBR was done once a week during the experiment period. Salt stress markedly reduced all tomato plant studied growth parameters, protein content, and increased contents of proline and lipid peroxidation. After treating tomato seeds with MF and applying foliar or spraying of hormone on tomato seedling leaves, significant enhancement and improvement in all studied tomato growth, yield, biochemical parameters and improvement in content of protein and proline occurred in plants grown under salt stress conditions. It is recommended to co-apply magnetic field with strength of 150 mt for 1 hour to tomato seeds with foliar application of 24-epibrasinolide, EBR, at level of 3 m. Keywords: salt stress, tomato, Lycopersicon Esculentum L, magnetic field, MF, 24-epibrasinolide, EBR, oxidative stress, SOD, APX, CAT, GR, proline, lipid peroxidation, MDA, mineral elements. Introduction. Salinity is one of the most important abiotic stresses, limiting crop production in arid and semi-arid regions, where soil salt component is naturally high and precipitation can be insufficient for leaching. Shrivastava and Kumar, 2015. Salinity stress is a term related to the accumulation of excessive concentrations of salts, especially NaCl in soil or water, and negatively affects the growth and development of many morphological and physiological characteristics of plants. Jamil et al., 2011. Salinity leads to ion toxicity followed by disruption of osmotic balance. NaCl in soil reduces absorption of water and K+ ions in plant cells, and increases accumulation of Na+ to toxic levels and produces reactive oxygen species, ROS, thus reducing plant growth and yield. Munz and Tester, 2008, Bajayerks Quintal et al., 2012. Magnetic field usually measured in terms of its magnetic induction B, whose unit is given in Tesla, T. Krylov and Tarakanova, 1960, were among the RST to report on magnetic field effects on plants. Magnetic fields augmented the germination percentage in many seeds, and the magnetic fields treated plants grow earlier than untreated ones. Shine et al. 2017. The auxin-like effect of MF was also suggested to explain ripening of tomato fruits. Bo and Solunka, 1963. Magnetic treatment was found one of the significant techniques known to reduce various abiotic and biotic stresses, including drought stress, Salim and El-Nadi, 2011, and salinity stress, Thomas, et al., 2013, Kutaria et al. 2017. Magnetic field is known to alleviate salt stress in plants without harming their environmental status, and at the same time influence the increase of yield in crops and improve organogenesis, growth, biomass accumulation under salt stress. Radhakrishnan and Kumari, 2013, Rathod and Anand, 2016, Buggel, et al., 2016. To counter the problems of salinity, Plants modify themselves by different means including detoxification of ROS through antioxidant enzymes and molecules, and induction of phytohormones, Munz and Tester, 2008, Ruiz-Lozano et al., 2012. Phytohormones are plant growth regulators which can mediate growth and development responses under ambient and stressful conditions, Peleg and Blumwald 2011. Growth and development in the sessile plants is regulated by the activity of some phytohormones like brassinosteroids, BRs, which control many physiological and biochemical processes, Iqbal et al. 2014. Endogenous application of phytohormones modulates physiological responses in plants that ultimately lead to adaptation, as is the case under salinity, Khan, et al., 2013, and exogenous application of phytohormones copes with salt stress, Iqbal, et al., 2012. Brassinosteroids, BRs, are a new group of phytohormones with significant growth-promoting properties and natural compounds of steroidal structure which have demonstrated to protect plants under saline stress. Brassinosteroids are involved in many other development processes, including flowering, leaf senescence, pollen development and male sterility, chloroplast development and root growth and development, UET al., 2011, YET al., 2010, Musig et al., 2003. The aim of the study is to evaluate the effect of tomato seeds pretreatment with magnetic field and foliar spray of brassinosteroids, hormone in reducing the negative impact of salt stress conditions in growth of tomato plant. 2 Materials and methods. 2.1 Effect of magnetic field, MF, treatments and foliar spray of brassinosteroids on tomato growth parameters under salinity. The experiment was conducted in pots, in the greenhouse of Department of Biological Sciences at King Abdulaziz University, Jeddah. KSA factorial design was used in this experiment. MF at strength of 50 mt, 100 mt, and 150 mt and foliar spray of brassinosteroids at different levels of 0.5 m, 1 m and 3 m were experimented on tomato growth parameters growing under different NaCl concentrations 50 mm, 150 mm, and 250 mm. The seedlings separated into 5 groups for different treatments as follows: 1. Control. 2. Seedlings under salinity 50, 150, 250 mm NaCl without MF or hormone treatment. 3. Pre-treatments with magnetic field divided into 9 subgroups of 50, 100, and 150 mt, watered with saline water. 4. Seedlings treated with Abeer hormone divided into 9 subgroups with 0.5, 1, 3 m concentrations, irrigated with saline water. And 5. Seedlings treated with both MF and hormone, and watered by saline water, to give a total of 0.8 pots and 618 plants. Plants were watered regularly with 1/2 strength Hoagland's nutrient solution 0, 50, 150 and 250 mm NaCl, respectively. 2.1.1 Magnetic field treatment. An electromagnetic field generator Testron M20 was used and seeds were primed with variable horizontal magnetic field strength, 50, 00 mt, and a gap of 5 cm between pole pieces, Vashist and Nagrajun 2008. The pole pieces were in cylindrical shape, with a diameter of 9 cm and 16 cm in length. The number of turns per coil was 3000 and the resistance of the coil was 16 ohms. A DC power supply, ADV/10A, with continuously variable output current was used for the electromagnet. The digital gauge meter, model DGM30 Testron, measured the magnetic field strength produced between the poles. The probe is made up of indium arsenide crystal and encapsulated to a non-magnetic sheet of 5, 4, 1 mm, it could measure 0 T with the full scale range in increments of 5 mt. The variation in temperature during the course of seed exposure was 25 degrees C. Under a uniform magnetic field, 100 seeds kept in the plastic container were placed between both poles of the electromagnet, shine, ETAL 2011. 2.1.2 Brassinosteroids treatment. The Brassinosteroids 24 APBL, 90% in purity, Pan Industry, Zhengzhou, China, was prepared by first dissolving 0.1 mg 24 APBL in 10 ml absolute ethanol followed by diluting the solution with milliq water ethanol, water, V/V, equals 1:10,000, AHAM DTAL 2013. The hormone was sprayed on tomato leaves at concentration of 0.5,1,3 m. 2.2 Soluble protein content. The total soluble protein content of the leaves was estimated following the method of Bradford, 1976. A 0.5 g of chopped fresh leaf material was homogenized in 5 ml of 0.1 m phosphate buffer. The homogenate was centrifuged at 5000 rpm for 10 minutes at 40 C.1.0 ml chilled 10% TCA was added to 1.0 ml of the supernatant, which was again centrifuged at 3300 rpm for 10 minutes. The supernatant was discarded and the pellet left was washed with acetone. It was then dissolved in 1 ml of 0.1 NaOH. To a 1.0 ml of aliquot, 5 ml of the Bradford's reagent was added and vortex mixed. The tubes were left for 10 minutes for optimal color development. The absorbance was then recorded at 595 nanometers on a UV-vis spectrophotometer, Bio20, Perkin-Elmer, Germany. The soluble protein content was quantified with the help of a standard curve prepared from the standard of bovine albumin serum, BSA. The protein content was expressed in mgg1fw. 2.3 Lipid peroxidation. The level of lipid peroxidation in the leaves was determined as malondialdehyde, MDA, content by the method of Heath and Packer, 1968. Fresh tissue of leaf was ground in 0.1% TCA and centrifuged at 10,000 rpm for 5 minutes. 1.0 ml of supernatant was taken in a separate test tube, to which 4.0 ml of 0.5% TBA was added. The mixture was heated at 95 C for 30 minutes. It was then quickly cooled in an ice bath and recentrifuged at 5000 rpm for 5 minutes to suspend the turbidity. The absorbance of the supernatant was read at 532 nanometers and 600 nanometers, corrected for unspecific turbidity by subtracting the value at 600 nanometers. The blank was 0.5% TBA reagent. The concentration of MDA was calculated using an extinction coefficient, DOT, of 155 mm1 cm1 as given in the following formula. MDA, nmol-G? FW, equals, A532, A600, XV/XW, X1000. Where, A532 equals absorbance at 532 nanometers, A600 equals absorbance at 600 nanometers, V equals extraction volume, W equals fresh weight of tissue. 2.4 Proline content. The proline levels of the leaves were estimated following the method of Bates et al., 1973. 500 mg of fresh leaf material were homogenized in 10 ml of 3% aqueous sulfosalicylic acid. The homogenate was centrifuged at 10,000 rpm for 10 minutes. 2 ml of supernatant, 2 ml of glacial acetic acid and 2 ml of acid ninhydrin were taken in a test tube. This mixture was incubated for 1 hour at 100 C to complete the reaction. The reaction was terminated by putting the tubes in ice bath. After the completion of incubation, the mixture was extracted with 4 ml of toluene. The extract was vigorously vortexed for 20 seconds. The chromatophore containing fraction was then aspirated from the aqueous phase and the absorbance determined at 620 nanometers using spectrophotometer. Bio20, Perkin-Elmer, Germany. L-proline was processed in the same manner to prepare the standard curve. The amount of proline was expressed as nmol-G. FW. 2.5 Statistical analysis. One-way NOVA using GraphPad Prism 6.0 software by Tukey's post hoc test, p 0.05. The experiment was carried out in triplicates, n equals 3, except for growth parameters, root and shoot length, fresh weight, dry weight, and RWC, where n equals 3. Two-way analysis of variance were used to examine the effects of two independent variables, factors, both individually and together. 3 results. 3.1 Effect of different concentrations of NaCl, MF strengths, and foliar spray of 24-epibersinolide on growth of tomato. Table 1 shows that tomato plants present high significant differences, p equals 0.01, in growth parameters, plant height, shoot and root fresh and dry weight, under NaCl, MF, and Abeer hormone different concentrations. Plant height, growth of shoot and root were significantly reduced under all NaCl concentrations, and highly significantly increased under all MF strengths and all levels of the hormone. Plant height decreased at rates of 15.82, 30.81, 42.99%, shoot fresh and dry weight significantly decreased at rates of 13.28, 45.89, 58.52% in fresh, and 41.56, 60.07, 74.45% in dry weight, respectively. Root fresh and dry weight significantly decreased at rates of 9.11, 40.02, 53.18% in fresh weight and 22.52, 58.36, 67.23% in dry weight, respectively under NaCl concentrations, 0, 50, 100, and 50, 250 mm. Where, plant height, shoot and root fresh and dry weight, showed successive and significant high, p equals 0.01, increases and improvement with application of MF and Abeer hormone. Plant height increased at rate of 84.59, 71.15, 85.86%, under MF and at rates of 76.38, 78.02, 87.2%, under hormone effect. Shoot fresh and dry weight increased at rates of 261.92, 262.92, 262.7% in fresh weight and 312.03, 320.81, 352.6% in dry weight. Root fresh and dry weights increased at rates of 220.77, 236.36, 212.23% and 190.62, 194.79, 210.41%, respectively under MF levels, 50, 100, 100, and 50 mt. And under Abeer, 0.5, 1, 3 m, sprayed on the leaves increase in shoot fresh weight reached 155.14, 185.81, 213.38%, and shoot dry weight 114.70, 131.16, 152.59%, while in root fresh and dry weight the increases were at rates of 152.03, 198.31, 242.33% and 92.7, 125, 150%, respectively at the different Abeer concentrations, 0.0, 0.5, 1, 3 m. It can be seen that the significantly highest reductions in all studied parameters occurred under 250 mm NaCl concentration. And the significant highest percentage increases of plant height was under 100 mt MF and 1 m Abeer hormone, and for shoot and root fresh and dry weight the highest values were under 150 mt MF and 3 m Abeer hormone. 3.2 Effect of interaction between different NaCl concentrations and foliar spray of 24-epibersinolide on tomato growth. Foliar application of the hormone Abeer gave successive and highly significant, P equals 0.01, improvement and enhancement in tomato growth parameters, plant height, shoot and root fresh and dry weights, under the influence of salinity with different NaCl concentrations, 0, 50, 150, 250 mm, Table 2. Application of the hormone Abeer at all rates, 0.5, 1, 3 m, significantly increased studied parameters of tomato plants under all NaCl concentrations, and increases of plant height reached, 34.34, 50.16, 74.43%, at 250 mm NaCl, shoot fresh and dry weight under Abeer 3m reached rates of 799.79, 1484.61%, respectively at 250 mm NaCl. In addition, in root fresh and dry weight increase in rates under 3m Abeer reached 1405 and 3550%, at 250 mm NaCl respectively compared to control without Abeer hormone. The increase in these parameters significantly increased with increasing NaCl concentrations to reach maximum percentage at 250 mm. The highest increase in plant height appear at 0 and 250 NaCl occurred under 0.5 m hormone, and under 3 m in the case of shoot and root fresh and dry WT. 3.3 Effect of interaction between different NaCl concentrations and strengths of MF on plant height, shoot and root fresh and dry weights, G, plant 1 of tomato. Magnetic field application gave successive and highly significant, P equals 0.01, improvement and enhancement in tomato growth parameters, shoot and root fresh and dry weights, under the influence of salinity with the different NaCl concentrations, 0, 50, 150, 250 mm, Table 3. At 50 mf plant height increased at rates of 49.5, 76.97, 124.7%, shoot fresh weight increased at rates of 209.22, 205.54 and 200.83%, shoot dry weight increased at rates of 210.74, 317.13 and 320.8%. Root fresh weight increased at rates of 155.84, 111.68 and 174.15%, while root dry weight increased at rates of 114.79, 133.33, 161.16%, respectively at NaCl concentrations, 50, 150, 250 mm. At 150 mf plant height increased at rate of 24.59, 85.05, 165.54%, shoot fresh weight increased at rates of 521.35, 441.25 and 397.74%, and shoot dry weight increased at rates of 244.44, 367.22 and 246.27%. Root fresh weight increased at rates of 211.03, 224.67 and 212.33%, and root dry weight increased at rates of 148.33, 208.33 and 154.58%, respectively at NaCl concentrations, 50, 150, 250 mm, compared to control. It can be seen that under control, the highest plant height was attained at 50 mtmf, and highest shoot fresh and dry weight was under 100 mtmf, while for root fresh and dry weight the highest values were under 150 mtmf. Moreover, at 250 mm NaCl highest increases were under 150 mtmf for all these mentioned parameters. 3.4 Effect of interaction between different strengths of MF and foliar spray of 24-ep Brassinosteroid on plant height, shoot and root fresh and dry weights, G, on plant 1 of tomato. Foliar application of the hormone Abeer gave successive and highly significant, p equals 0.01, improvement and enhancement in tomato growth parameters, shoot and root fresh and dry weight, at all MF strengths, 50, 100, 100, and 50 mt, Table 4. At 50 mf plant height increased at rates of 70.84, 92.36, 90.57%, shoot fresh weight increased at rates of 249.22, 255.54, 280.83%, and shoot dry weight increased at rates of 270.74, 337.13, 330.8%. While root fresh weight increased at rates of 205.84, 211.68, 244.15%, and root dry weight increased at rates of 194.79, 183.33, 191.16%, respectively at levels of Abeer hormone, 0.5, 1, 3 m. At 150 mf plant height increased at rate of 82.04, 91.66, 83.89%, shoot fresh weight increased at rates of 821.35, 841.25, 797.74%, and shoot dry weight increased at rates of 344.44, 367.22 and 346.27%. Root fresh weight increased at rates of 211.03, 224.67, 212.33%, and root dry weight increased at rates of 208.33, 208.33, 214.58%, respectively at levels of Abeer hormone, 0.5, 1, 3 m, compared to control without Abeer hormone. It can be seen that the highest increase in tomato plant height was attained at 100 mt/mf with 1 m Abeer hormone, and for shoot fresh and dry weight under 100 mt/mf at 3 m Abeer, while for root fresh and dry weight it was at 150 mt/mf with 1 and 3 m Abeer respectively. 3.5 Effect of interaction between different NaCl concentrations with magnetic field and foliar spray of 24-epibrasinolide on growth parameters, plant height, cm, shoot length cm, root length cm, leaf area cm2 on tomato. The results in Table 5 illustrate significant differences in the interactions between effects of NaCl concentrations, MF magnitudes, and the hormone Abeer on tomato growth parameters, plant height, shoot and root fresh and dry weight per plant, gm. MF and Abeer hormone application resulted in significantly high percentage increasing growth parameters in tomato under all NaCl concentrations, 0, 50, 100, and 50, 250 mm. Under 50 NaCl concentration the maximum value percentage were 49.97%, in plant height under 100 mf and 1 m Abeer. Shoot fresh weight was 215.59%, under mf 150 t and Abeer 1 m, in shoot dry weight, 298.07%, under mf 150 t and Abeer 1 m, root fresh weight, 195.51%, at mf 150 t and Abeer 1 m, root dry weight, 150.13%, under mf 150 t and Abeer 3 m. Under 150 NaCl concentration the maximum percent increase in plant height was 29.09%, under 100 mf and 3 m Abeer, shoot fresh weight was 1446.11%, under mf 150 t and Abeer 1 m, in shoot dry weight, 516.44%, under mf 150 t and Abeer 3 m, root fresh weight, 260.16%, At MF 150T and EBR 1M, root dry weight, 385.13%, under MF 150T and EBR 3M. Under 250 NaCl concentration the maximum percent increase in plant height was 180.93%, under 150 mF and 1M EBR, shoot fresh weight was 350.89%, under MF 50T and EBR 3M, in shoot dry weight, 516.44%, under MF 50T and EBR 1M, root fresh weight, 260.16%, at MF 150T and EBR 1M, root dry weight, 4,555.14%, under MF 150T and EBR 1M. The highest percentage increases in plant height and shoot and root weight were attained by the hormone at the highest salinity stress 250 mm under all MF strengths. It seems that the effect of the hormone was more pronounced under high salinity rate than under low rates. EBR hormone significantly enhanced tomato shoot and root growth and alleviated effect of salinity under the highest NaCl concentration 250 mm compared to the lower NaCl concentrations. 3.6 Effect of different NaCl concentrations with MF strengths and foliar spray of 24 epibrasinolide on number of leaves plant 1, leaf area, number of flowers and number of fruits plant 1, on tomato. Table 6, shows that tomato plants have undergone high significant differences, p equals 0.01, in growth parameters, number of leaves plant 1, leaf area, number of flowers/plants, number of fruit/plant, under NaCl concentrations, MF effects, and EBR different concentrations. Number of leaves/plant reduced at rates of 24.07, 33.70, 49.38%, leaf area reduced at rates of 17.83, 32.14, 47.22%, flowers/plant significantly decreased at rates of 2.04, 21.92, 43.6%, fruit/plant reduced at rate of 7.82, 32.63, 44.34%, respectively under salinity stress of 50, 100, and 50, 250 mm compared to control. All these parameters significantly highly increased, p equals 0.01, with increase in MF, 50, 100, 150 mt. Number of leaves/plant increased at rate of 104.73, 108.64, 97.18%, and leaf area increased at rates of 136.50, 143.34, 1219.65%, number of flowers/plant reached 112.63, 136.70, 128.49%, and number of fruit/plant 164.44, 156.09, 172.04%, respectively at the different MF, 50, 100, 150 mt, compared to control without MF. All these parameters significantly increased, p equals 0.01, with increasing concentration of EBR, 0.5, 1, 3m, sprayed on the leaves. Number of leaves/plant increased at rates of 100.0, 100, and 1.23, 108.78%, and leaf area increased at rates of 128.13, 133.16, 147.9%, number of flowers/plant reached 47.88, 70.56, 70.56%, and number of fruit/plant reached 81.23, 100.09, 115.57%, respectively at the different EBR concentrations, 0.5, 1, 3m, compared to control without EBR hormone. It can be seen that MF application at 100 level gave the highest significant increases in number of leaves/plants, leaf area, and flowers/plant, compared to 50 and 150 levels, while MF 150 exceeded in number of fruit/plant. As for the hormone, concentration 3M dominated, giving the highest increases of all these studied parameters compared to the other concentrations. 3.7 Effect of interaction between different NaCl concentrations with magnetic field on number of leaves plant 1, leaf area, number of flowers and number of fruits plant 1, of tomato. Application of MF highly significantly, p equals 0.01, improved and enhanced tomato growth parameters, number of leaves/plant and leaf area, number of flowers/plants, number of fruits/plant, under salinity of NaCl concentrations, 0, 50, 100, and 50, 250 mm, Table 7. Application of MF at strengths of 50, 100, 150 mt, significantly increased all parameters under all NaCl concentrations. Under 50 mm NaCl percentage increase in number of leaves/plants reached 67.79, 64.11, 87.06%, and leaf area, 100.94, 109.17, 121.89%, and number of flowers/plants reached 106.95, 140.50, 117, 89%, fruit/plant, 179.43, 164.78, 199.03%, under MF, 50, 100, 150 mt, respectively. Moreover, under 250 mm NaCl concentration number of leaves/plants increased and reached 666.06, 800.36, 800.36%, and leaf area, 308.07, 331.42, 295.53%, number of flowers/plants reached 142.00, 153.3, 156.6%, number of fruit/plant, 130.94, 152.65, 161.66%, under MF 50, 100, 150 mt respectively compared to control without MF. Application of MF ameliorated the harsh salinity impact on tomato number of leaves/plants, leaf area, number of flowers/plants, number of fruits/plants, particularly number of leaves/plants, leaf area, number of flowers/plants at all NaCl concentrations 50, 100, and 50, 250 mm. The best MF strength is 100 and 150 mt. 3.8 Effect of interaction of different NaCl concentrations with foliar spray of 24 Brassinosteroid on number of leaves plant 1, leaf area, number of flowers and number of fruits plant 1, of tomato. Foliar application of the hormone Abeer gave successive and highly significant, p equals 0.01, improvement in tomato growth parameters, number of leaves/plant, leaf area, number of flowers/plant, number of fruits/plant, under salinity of different NaCl concentrations, 0, 50, 100, and 50, 250 mm, and the lowest values were at level 250 mm NaCl, Table 8. Application of the hormone Abeer at all rates, 0.5, 1, 3m, significantly increased all these parameters of tomato plants under all NaCl concentrations. Under control percentage increased in number of leaves number/plant reached 39.81, 52.92, 544.17%, and leaf area increased at rates of 53.75, 77.72, 190.17%, flowers/plant reached 34.34, 57.43, 43.58%, number of fruit/plant reached, 105.43, 124.33, 132.44%, under EBR levels of 0, 0.5, 1, 3 m respectively. Under 250 mm NaCl concentration increased in leaves number/plant reached rates of 40.12, 51.02, 564.22%, and leaf area increased at rates of 73.36, 72.46, 223.21%, number of flowers/plant reached, 56.6, 80.0, 93.3%, number of fruit/plant, 42.37, 74.36, 88.56%, under EBR levels of 0.5, 1, 3 m respectively compared to control without EBR hormone. Hormone application enhanced and increased all these studied parameters, with hormone concentration 3 m dominating at all NaCl concentrations. 3.9 Effect of interaction between different MF strengths with different concentrations of foliar spray of 24-ep Brassinosteroid on number of leaves plant 1, leaf area, number of flowers and number of fruits plant 1, of tomato plants. Foliar application of EBR hormone gave successive and highly significant increasing, p equals 0.01, in tomato growth parameters, number of leaves plant 1, leaf area, number of flowers/plant, number of fruits/plant, together with the influence of the different MF strengths, 50, 100, 100, and 50 mt, table 9, and parameters increased with increase in hormone concentration levels and with MF strengths. Under MF 50% increase in number of leaves/plant increased at rates of 93.95, 107.50, 112.77%, and leaf area increased at rates of 114.06, 135.73, 159.31%, flowers/plant reached 96.20, 111.41, 129.5%, number of fruit/plant reached 138.36, 152.43, 193.15%, under EBR levels of 0.5, 1, 3 m respectively. And under MF 150% increase in number of leaves/plant increased at rates of 95.77, 92.70, 102.25%, and leaf area increased at rates of 128.51, 127.75, 132.69%, flowers/plant reached 116.36, 142.64, 127.92%, number of fruit/plant reached 172.04, 173.54, 170.26%, under EBR levels of 0.5, 1, 3 m respectively compared to control without EBR hormone. It can be seen that highest increase in number of leaves/plants, leaf area, number of fruits/plants under 50 mt/mf with 3 m hormone level, while number of flowers/plants was at 100 mt/mf with 3 m. 3.10 Effect of interaction between different NaCl concentrations with magnetic field and different concentrations of foliar spray of 24-ep Brassinosteroid on number of leaves plant 1, leaf area, number of flowers and number of fruits plant 1, of tomato plants. The results in table 10 show that application of the hormone EBR sprayed on tomato leaves resulted in high significant increases in growth parameters, number of leaves/plant, leaf area, number of flowers/plant, number of fruits/plant, under all interactions with MF strengths, 50, 100, 100, and 50 mt, and salinity stresses represented in NaCl concentrations, 0, 50, 100, and 50, 250 mm. Application of the hormone and MF mitigated the negative effects caused by NaCl salinity on these studied parameters, and at the same time, these enhancement effects are gradually decreased with increase in salinity up to 250 mm NaCl. Under control, the highest percentage increase in number of leaves/plants was 86.78% at 3 MEBR and 100 mtmf. Leaf area was 131.71% at 3 MEBR and 50 mtmf. For flowers/plant was under 1 MEBR hormone level and 100 and 150 mtmf, 107.75%. The highest increase in number of fruit/plant was at 3 hormone level with 50 mtmf, 218.51%. 3.11 Effect of different concentrations of NaCl stress, magnetic field, and different concentrations of foliar spray of 24-epibrasinolide on biochemical parameters, soluble proline content, lipid peroxidation, and proline content, on tomato. Tomato plants have undergone high significant differences, p equals 0.01, in the biochemical parameters, soluble protein content, lipid peroxidation, and proline content, under NaCl, MF, and EBR hormone different concentrations table, 11. Soluble protein content was significantly reduced with increase in NaCl concentrations, while lipid peroxidation and proline content were significantly increased with increase in NaCl levels, 0, 50, 100, and 50, 250 mm. On the other hand, application of MF and EBR hormone caused significant increases in soluble protein content, and significant reduction in lipid peroxidation and proline content under all MF strengths, 50, 100, 100, and 50 mt, and hormone levels, 0, 0.5, 1, 3 m, compared to controls. The significantly highest reduction in soluble protein content, 50.44%, and the significantly highest increase in lipid peroxidation, 147.28%, and proline content, 107.8%, compared to control were under the highest NaCl concentration, 250 mm. Application of MF and EBR hormone significantly enhanced and increased soluble protein content, and reduced lipid peroxidation and proline content. The significantly highest increase in soluble protein content, 701%, was at MF 100 mt, highest reduction in lipid peroxidation, 30.09%, was at MF 150 mt, and in proline content, 15.42%, was under MF 50 mt. The significantly highest increase in soluble protein content, 73.88%, and the significantly highest reduction in lipid peroxidation, 24.87%, and proline content, 22.6%, compared to control, were under the highest application of EBR hormone at 3 m levels sprayed on the leaves compared to control. For discussion. Results obtained on reduction of plant height, shoot and root fresh and dry weights by salinity in this research agree with other research findings. Salt stress caused significant reduction in sugar beet plant biomass, Ghulam et al., 2002, Ajwain, Ashraf and Oroi 2006, Menthopulegium, Oues Lati et al. 2010, Cumin, Rebbe et al. 2017, and in Schizonepetitinuifolia, Joe et al. 2018. This decrease in growth under salinity may be because salinity affects stomatal closure and ultimately photosynthetic activity is reduced, Tarat et al. 2012, and causes nutritional disorders, Lochley and Epstein, 1990, Anuradha and Rao, 2003. Plant dry weight significantly decreased by increasing NaCl concentration, Khalid and CAI, 2011. Atok et al., 2003, applied MF on soybean and corn and found significant increases in fresh and dry weight of the seedlings. Application of MF enhanced growth and yield of wheat, barley, maize, soybean, and mung bean under abiotic stresses like salt and drought. Hossein et al., 2018. Martinez, et al., 2000, exposed barley seeds to a static magnetic field of 125 mt for different periods, and observed increases in lengths and weights of seedlings. Kataria et al., 2017, reported similar results. The role of MF may be it causes an increase in the absorption of water and nutrients. Al-Hassan et al., 2011, or MF might send signals for protein synthesis immediately after the salt stress that protects the plasma membrane. Santoro et al., 1992. Karami et al., 2012, found that MF adjusted osmotic process and improved water availability to plants. As for the role of EBR hormone, Dong, et al., 2017, suggested that it works on the root by increasing root activity and H plus AT-PASA activity. Moreover, Kripak et al., 2000, have demonstrated that EBR could regulate cell elongation and division by activating cell wall loosening enzymes. Lopez Gomez et al., 2016, demonstrated that foliar application of 24-EBL increased plant fresh and dry biomass of Medicago truncatula under saline conditions, and in strawberry, Carladag et al., 2011. The results obtained regarding number of leaves/plants, leaf area, number of flowers/plant and number of fruits/plants are compatible with research findings by others. The immediate response to salt stress is a reduction in the rate of leaf area as the salt concentration increases. Wang and Nil, 2000. 80% reduction in plant growth under high salinity is due to the reduction of leaf area, La, and 20% of the growth reduction is most likely due to decrease in stomatal conductance. Parada and Das, 2005. In addition, Bugel et al., 2016, found significant increase in leaves number and leaf area of soybean, and, Bugel et al., 2019, in maize when subjected to MF. After application of the hormone EBR there were significant increases in number of leaves/plant and leaf area in a number of plant species. Triticum aestivum, Schaubas et al. 2008, Capsicum annuum, Haueriumli et al., 2010, Solanum melangina, Wu et al., 2012, Cajanus Cajun, Dalio et al., 2011, Phaseolus vulgaris, Radi, 2011, Zia maize, Agami, 2013. Selic et al., 2008, mentioned that MF activation comes from the fact that magnetic field affected cell division and protein synthesis in Paulownia node cultures. Maheshvari, 2009, said that magnetic treatments may affect phytohormone production causing increase in plant growth and cell activity. Blitz and Gallagher, 1991, found reduction in the number of inflorescences due to salinity effects. Flowering is delayed in wild mustard, Sinope arvensis, and in Arabidopsis. Stanton et al., 2000, Accard et al., 2006, as effect of salinity. Flowering of rice is delayed by salinity, even at low NaCl concentrations, 20, 0 mm, Lutz et al., 1995. Cariouti et al., 2007, and Megan et al., 2008, reported reduction in total yield of tomato at salinity. Megan et al., 2008, found reduction in tomato fruit number of 2% with an increase of 1 dsm1 beyond the threshold. Haji Boland et al., 2010, said that salinity effects on tomato growth and yield reduction might be due to variation in photosynthetic products translocation toward root, decrease of leaves, partial or total enclosed of stomata, direct effect of salt on photosynthesis and ion balance. EBR hormone improved activity of enzymes included in photosynthesis, thus increasing number of flowers and number of fruits/plant under salt stress. Xia et al., 2009, Chen et al., 2015. Number of researchers observed reduction in protein content in plants after treatment with sodium chloride, in tomato, Jalil et al., 2008, in barley Hortum vulgar, Khosravinjad et al., 2009, and in Vigna unguiculata, Chen et al., 2007. Aurora et al., 2008, found that EBR application enhanced protein content in Zea maize seedlings under salinity stress. The promoting effects of EBRs on the protein content could be attributed to activation of transcription and translational processes of specific stress tolerance genes, Kagel et al. 2007. Bilyevskaya, 2004, concluded formation of new protein bands in plants treated with magnetic field. Lipid increased in plants under salinity because lipid is formed as a result of oxidative damage that takes place in the tissues of plants suffering salt stress, Campo et al., 2014. Egbici et al., 2013, reported that salt stress induced oxidative damage to membrane lipids. Magnetic field application initiates production of radicals acting as a signal to enhance production of antioxidants and cause a dramatic increase in radical scavenging activity, thus preventing oxidative damage due to salinity and reduces lipid, Saheb Jameh et al., 2007, Selik et al., 2009. Chen et al., 2011, reported that MF reduces lipid peroxidation. The rise in proline content in tomato under salinity is because salt stress causes a remarkable increase in free proline content in plants, because it helps in alleviating the osmotic stress upon cell under stress conditions by reducing the uptake of toxic ions, and it interacts with cellular enzymes and stabilizes their structure as well as functions, Hayat et al. 2012. The accumulation of proline in plants under salinity stress is caused either by the induction of expression of proline biosynthesis genes, P5CS, P5CR, or by the repression of the genes of its degradation pathway, PDH silencing, Marco et al. 2015. Application of EBR on leaves resulted in rise of proline in leaves and helped plants to tolerate salt stress, as in pepper plants, Abbas et al., 2013, Cucumis sativus plants, Ferdudin et al., 2013, P. vulgaris seedlings, Rady, 2011, and in Triticum aestivum, Talat and Shaki, 2013. V. Conclusion. The results obtained in this study documented that salinity exposed negative effects on morphological, physiological, and biochemical parameters of tomato plants, initiating reactive oxygen species, ROS, in the cells of the plants, thus causing oxidative stress, increasing antioxidant enzyme activities. Salt stress induces osmotic stress, ionic stress, and oxidative stress and reduce tomato growth and yield. Application of magnetic field on tomato seeds and spray of 24-epibrasinolide hormone on tomato leaves significantly positively affected and improved all these mentioned tomato parameters and made tomato plants more tolerant to effects of salt stress. As a result of application of MF and EBR hormone on tomato growing under salinity stress conditions, there were significant increases in plant height, shoot and root fresh and dry weight, number of leaves/plant, leaf area, number of flowers/plant, number of fruits/plant, increase in protein content in leaves, decrease in lipid and proline. Resistance to any kind of stress closely linked to detoxifying ability of plants to counteract the ROS. There is a close link between ROS and MF treatment. Our results indicated that MF has a dual beneficial impact on ROS. MF enhanced the ROS and later reduced the ROS in mature leaves to facilitate higher electron transport, efficiency of photosynthesis, soluble portion, MDA, and proline content, which ultimately results in higher yield of tomato under salt stress. In addition, application of 24-epibrasinolide hormone mitigated harsh and inhibiting effects of salt stress on tomato plants. Thus, The present study provides the evidence that MF pretreatment of seeds and foliar spray of EBL can enhance the tomato plant salt tolerance. MF seed pretreatment can be used in agriculture to obtain better growth and increased yield under adverse abiotic stress conditions. Pre-exposure of seeds to MF has potential to increase crop production per unit area of land without having any detrimental effects toward any environmental component. In future, it may be a good tool for improving agricultural production in several economically important crops since it mitigates the adverse effects of abiotic stresses. In addition, the present study revealed the ability of Brassinosteroids hormone in improving the yield of tomato plants under salt conditions. It has been used extensively in agricultural uses. In addition, foliar application of these plant growth substances may be more cost-effective as it requires relatively lower concentrations compared to other substances.